Hi everyone. So my name is Carlene, Blailes, and I'm Palawan from Guam, born and raised in Guam and moved to Hawaii for college. So my research project today is understanding lizards in our ecosystem. So understanding lizards, before the whole science, let's talk a little bit on the history of lizards and its history in Hawaii. So, there you go. Okay, so starting off, we want to talk about Hawaii wahine. I think it's a name familiar with most of you folks here in Kauai Nui. Lizards back in ancient times, they were known to be protectors of large bodies of waters. And Kauai Nui being one of the largest fish ponds here in Kailua, or here in Hawaii, Hawaii wahine is the protector of this place. And she is a shape-shifting deity. She's known to shape-shift between a beautiful Hawaiian lady and a big giant lizard, as you can see in the picture here. And she's known to protect the fish pond by making sure that these fishermen don't get too greedy and take all the fishes. And so yeah, and her presence when she's there, you can tell because the grass and the leaves, they'll turn golden color in her presence, and when she's gone, it turns back to green. So lizards. So in my project, I'm studying 3 lizard species here in Hawaii. The first one is the brown Anolis lizard, the Anolis sagrei. There's a little picture of it. The Anolis carolinensis, the green Anolis lizard, and the Phelsuma laticauda, the day gecko. But did you know that here in Hawaii, lizards are not native to the island? Instead, they were all introduced. And for my 3 species, for the brown Anolis, they're native in Cuba. The green Anolis, they were native in southeastern North America before they got introduced to Hawaii. And the Phelsuma laticauda, the day gecko, they are native in Madagascar and actually has been recently introduced to Hawaii in the 1970s, which was really quite recent. So getting into what we're studying in the lizards, we're studying the microbes and parasites interaction. We're studying the nematodes. Nematodes are little parasitical worms found inside reptiles, hence why we're studying in lizard. There's a little picture of how they look like. You'll see it more. Just a little warning, if you guys are not into worms, you're gonna see pictures of worms today. So the objective here is understanding the gut bacteria and the gut parasites. So we're studying the guts of the lizards. So these microbes and parasites, they can affect both humans and animals. They can affect the host's health. For example, for parasites, they can affect brown Anolis lizards. A recent study just popped up that with this parasite, they affect the growth of these lizards, making them more smaller. For human health, there is actually a recent study of a clinical treatment for this disease called Clostridium difficile. So it's a clinical treatment is known as the fecal microbiome transplant because this disease there's no antibiotics that can work with it. So this treatment is basically a microbe and parasite interaction to help with this disease, which is inflammation in the bowel movement, and to help cure it. And there has been recent studies on how this treatment can possibly help with treatments with maybe cancer and also bipolar disorder symptoms. Okay, so moving on this project, we had 3 hypotheses. The first one being that nematodes increase bacterial diversity. And as you can see here, we made an experimental design that with more worms found in the gut, there'll be more bacterial diversity, and that the worms itself is affecting the bacteria. The second one is kind of the opposite. Maybe that bacteria affects the worms, so that with bacterial diversity, it can reduce the risk of parasitic infection. That all means that with more nematodes found, there'll be less bacterial diversity. Perhaps these bacterial species are gonna take the resources that needed for the worms to grow, so that makes them lessen. The last one is based on the lizard species. Maybe there's no interaction between the microbes and parasites, but instead maybe the lizard species is the one that is the main cause of all of it. Maybe with lizard species, some will have more nematodes. Maybe there'll be different nematode species in each lizard species. So how did we do this? So this was conducted in the lab. We did a collaboration with Dr. Amber Wright's lab. Her lab is in Manoa. She's a researcher who mainly studies lizards. So we used her samples, and she collected these samples in Kanyohei, and these samples that we collected, or the samples that she collected dated back between 2018 and 2019. So what we first did is look it under the microscope. So these samples that we have are just purely the gut, like it's already separated from the lizards. And what we did is that we opened the gut up, we count how many nematodes are there, we jot it down in a little table, and then we run it into a DNA extraction. We try to beat down these nematodes until only the DNA is there, and then we run it through a PCR. And as you can see here, there's me doing the gel. We run through a PCR, we put it inside a little agarose gel that allows us to see the DNA in a different light. And then we conduct DNA sequencing. So from the samples, we got 14 samples from the brown anoles, 11 samples from the green anoles, and 17 samples from the day gecko. So, oh yeah, there's more morning now. So PCR didn't work. So we didn't get to know what types of nematode species they were. So we had to categorize them in morphospecies based on what we can observe from them. So the first one that we saw here, I had to circle it because it's so small, you cannot see it. They're like microscopic worms. So these ones are found in the stomach of the lizards. We call them morphospecies A. The second one is morphospecies B. These ones are way, way bigger. You can actually see them without even looking in the microscope because they're that big. They range between, I think, 1 to 2 millimeter. And these were found in the stomach of the lizards. Similar to morphospecies A, these worms are more smaller. You cannot see them, you have to look under the microscope, but these were found in the intestines of the lizards. Now, morphospecies D. So these ones were also found in the stomach of the lizards, but you can see here that there's actually like little stripes around them. So we consider them to be a whole different species compared to the other three. Now, morphospecies E. This one is a funny story because one day I was looking at it, I was trying to dissect, and there was nothing in the stomach. And all I found was these lesions around the stomachs here. And I grew curious, so I poked at one of them and popped all these like little worms inside it. So if I didn't discover that, I probably would have thought that that lizard sample did not have any worms. So I found that pretty interesting. So what did we find out from these? So our first figure here shows that the brown anoles has the most worms. As you can see here, so based on that, the Anolis sagrei, the brown anole, was a lot significantly different compared to the other two due to the amount of worms they had. And the Phelsuma laticauda, you cannot really tell, but out of the 17 samples, we only found one sample that only had one worm. So the worms were located mainly in the stomach, as you can see in the blue. So these colors represent the location. Blue being the worms in the stomach, red being worms in the intestines, and yellow being worms outside the stomach, the lesions that we saw in morphospecies E. And as you can see here in the green anole, those were mainly the worms outside the stomach. So for the morphospecies, we categorize them. And as you can see here, that there's morphospecies that changes between lizard species, with Anolis sagrei having morphospecies A, B, and C, and the green anole having actually all of them A to E, but D and E are more, you can find them more in that green anole compared to, but not in any of the two. For the day gecko, we only found one sample, and that was morphospecies A, which was worms located in the stomach. So we wanted to see the lizard lengths, comparison between the lizard lengths and the total worms. We did a little log 10 of the total worms due to the varying amount of data we had or numbers we collected. So based on this, you can see that there's a trend line going down that suggests that with more worms found, the smaller the lizards. However, we ran a little p-value to see if it's significant. Unfortunately, it's not significant, meaning that there's actually no effect that it had with each other. And then the bacterial richness has no effect on the total worms per lizard. Despite having the trend line here, we did run a p-value again, and it still had no effect to it. But however, we also did the bacterial diversity. a comparison between the total worms and the bacterial diversity. Bacterial diversity is basically the different species of bacteria, but in like it ranges on how even these species are, which is a little different between bacterial richness, which is just like the species itself. So for the bacterial diversity, despite having the trend line going down, which suggested that maybe bacterial diversity reduces worms. However, we did do the data analysis to see if that's really significant, and because of our varying samples and total worm data is being so varying in size or numbers, it's not really that significant. So what does that all mean? In summary of all that graphs and charts you saw, nematodes do differ between lizard species. However, the microbiome did not determine parasitic infections. But there's still chances that maybe we missed something. For example, we didn't do the PCR. We didn't get results. So maybe knowing these types of nematode species, we can learn more about like what's really happening inside the lizard. And secondly, maybe the bacterial species, there's certain ones that does affect it, but we didn't get to run enough information on it. So in conclusion, coming back that lizards are not native here in Hawaii, it's still important to understand lizards in our ecosystem because they're now here in Hawaii. They're now common. You see them in your house, you see them everywhere. So it's important to understand these lizards as they are kind of invasive species, and invasive species comes invasive parasites. And how understanding the microbiome can also affect the stability of like the conserving native species, such as like how the honey creepers, as Mahena said, are affected by diseases. It can help with probably learning to conserve these species. So these are my acknowledgements. I want to thank my mentor and Kumarmat, Kumakayana. Without them, this wouldn't be possible. And thank you for everyone for being here and listening to my presentation.